Hi, I am Adelaide Miarinjara, and I'm a scientist at the Department of Environmental Science. I am from Madagascar. I grew up there and did all my education in Madagascar. And when I came to the United States, it's to start the postdoc program. My interest in science started at a very young age, I think, because I was quite curious about everything surrounding me. And that's why I started a biology courses at the university in Madagascar. I got my PhD in 2017, and after that, I was starting to look for something more beyond Madagascar. So I applied for several postdocs, and my first postdoc was at the Joe Hinnebusch lab at NIH in the lovely town of Hamilton in Montana. So my first contact with United States happened in a very beautiful place in Montana at the Rocky Mountain Lab. My research interest is around disease transmitted by insect. In fact, my training is medical entomology, and I am still working on bubonic plague, a disease transmitted by the bites of fleas. When talking about bubonic plague, we always think about of a disease of the Middle Age in Europe, the Black Death or the Plague Doctor, and that's for me is a really familiar assumption. But the disease is doing well and still present in the world, in several countries, mostly in Madagascar, where all my research is focused.
So to unpack that slightly further, could you maybe start by highlighting what aspects of biology first drew you to the field, and maybe what your PhD was exploring?
So my PhD was really around bubonic plague, vector control, in fact. So because bubonic plague is a disease transmitted by insect, vector control is a big part of the things the government will do to try to avoid the spread of the disease. So vector control means the use of different devices or means to reduce the number of fleas and then reduce the contact between people and fleas.
And so I think you mentioned elsewhere that other model systems are often quite common in Malagasy research, so particularly looking at lemurs. How does your research vary from that?
So Madagascar is quite a unique ecosystem. I saw recently that 5% of species worldwide are present just in Madagascar, that small island east of the African continent. So that's amazing regarding the size of the island. So it is really important that people work on conservation, and lemur is quite an iconic animal for biodiversity and conservation in Madagascar. But as far as I am concerned, I am not working on conservation, even if I know that there is a link between the natural world, conservation, and the spread of zoonotic disease to the people.
And maybe for a broader audience, could you describe what zoonotic diseases are?
So zoonotic diseases are diseases from animals that are going or jumping into people.
And so I think after, of course, we've just had a COVID pandemic, we realised of course we're still trying to determine the origins of SARS-CoV-2. But I think the risk of future zoonoses is particularly concerning. How do you imagine your research, particularly understanding vector-borne diseases and their applicability to other vector-borne diseases, might have broader relevance as well?
So vector-borne diseases are becoming more and more important, I think, because of the ongoing climate change and also the challenges faced by the growing number of, let's say, human beings on Earth. So spanning from the availability of resources, competing for territories with like natural environment. So those change in our landscapes are influencing the diversity and the distribution of those potential vectors, and will certainly have an impact on the spread of zoonotic and vector-borne disease.
Yeah. And I think maybe in some ways Madagascar could be considered an eighth continent if you just go by animal and plant diversity, as you mentioned. Is the idea therefore that because Madagascar has such a rich and interlinked ecosystem that disruptions, and would you say that perhaps Madagascar is among the countries that's most gravely affected by climate change? It's particularly sensitive to disruptions caused by climate change, and this is what could allow zoonoses and also more opportunities for plague transmission.
Regarding plague in particular, regarding what we know about plague, it is quite difficult for now to make like a direct assumption about the potential distribution of plague, for example, in the future, since the disease is favored, let's say, by a more temperate climate. And sometimes when we think about climate change, we think about like the rise of temperature. So the rise of temperature may change plague distribution in Madagascar.
Certainly, and I think particularly where in other parts of Africa we're already seeing the effects of increased transmission zones of Anopheles and Aedes mosquitoes that also carry arboviruses. How is the geography of Madagascar particularly likely to change over the course of the next few decades with climate change?
So, with climate change, because Madagascar is an island, so with the rise of the level of the oceans, we will certainly have a lot of problems with all of the coastal areas. Even now, we are starting to see villages flooded by high tides. So that means a lot of displaced people. And we already know from the lessons from the past that displaced people means like people being exposed to new vectors, new diseases, people not not exposed to other disease before, so not having the immune system to withstand those diseases. So that means also like concentration of people in smaller area. So I think the biggest challenge for Madagascar is the rise of the sea level and also drought.
Yeah. And of course, Madagascar has seen a lot of human-induced anthropogenic changes that had happened over the course of its history. Have you also seen changes in vegetation patterns? I know that Madagascar has a lot of shrubland, but also dense forest on the eastern coast. Are these patterns also changing? And are many of the animal-borne diseases in Madagascar linked to particular areas of this Greenland or shrubland?
So, for plague in particular, there are few things that still to be discovered, but there are some hints of possible existence of plague in the wild small mammals in the highland forests. So people's encroachment within the forest area might expose them to plague transmission in that like sylvatic cycle.
Yeah.
But most of the time, plague epidemics are happening in villages, like a bit, let's say, far away from dense forest in those shrublands. Something that is always brought up is the bushfire. It is said that the people are burning the bushes, and let's say the rats living in those bushes are going toward the villages, so the rat density would increase with the bushfires, and then exposing people more and more with disease such as plague.
I see. And I guess this is also a recurrent theme in the lab you're currently working with. This is the Gillespie lab.
Yes.
Would you maybe want to describe what you do and what the Gillespie lab is involved in more broadly?
So the Gillespie lab also has work about like zoonotic diseases, transmission between human, disease from human that can potentially transmit it to wildlife.
Yeah.
Also, like the spread of antibiotic resistance within wildlife and natural world, which is such a big concern.
Yeah.
In Madagascar, the Gillespie lab is also working on the diarrheal diseases, and me in particularly now I am interested in flea infestation inside households.
And so, I mean, maybe before, I think broadly, what people, of course, are familiar with the plague as you've described as a ravaging disease of the Middle Ages of the 14th century, and some of the repercussions during the Roman period. But maybe could we perhaps explore in more detail what causes the plague, some of the symptoms of the plague. So maybe if we start with that first part, what's the pathogen underlying the plague, and maybe could we describe some of its life cycle?
Yes, so when we talk about bubonic plague, we think about Yersinia pestis, the agent, which is a bacterium. This bacterium evolved to be able to be transmitted by flea bite, meaning that in its life cycle it needs to be inside the flea gut to be able to be transmitted. You know probably also the pneumonic plague, another form of the plague, but it's like a secondary form stemming from bubonic plague. But when the plague is reaching the pneumonic form, it's more dangerous because it doesn't need fleas anymore to carry it around, but human-to-human contact. So one example of that, like how terrifying pneumonic plague was, what happened in Madagascar in 2017, where urban outbreak of pneumonic plague happened. So the most striking symptom of bubonic plague is the sudden high fever, fatigue, and the appearance of a bubo, which is the lymph node totally invaded by the bacterium Yersinia pestis. So the bubo is just the body trying so hard to stop the infection at the lymph node, and the lymph node is sore, and that's the bubo. It's very painful.
And it appears like a swollen extension, effectively. And as far as we know, this is the only Yersinia pestis is the only bacterium that actually causes buboes to rise in the body.
Yeah, I think a bubo is really different from any growth, because as far as I know, when you have like infection somewhere, you may have like swollen lymph nodes. If you have a cut, for example, infected cut on your legs, you may have a growth on your groin.
Yeah.
But for Yersinia pestis, it's the growth is really infected. Sometimes it can break. Yeah.
So I'm curious, do you therefore expect that, at least for the pneumonic form of the bubonic plague, if it's a secondary cause, that the most typical route transitions from bubonic to pneumonic in a population, and that most of the time, because it's an encounter with, in this case with wildlife, that it doesn't reach the stage of becoming pneumonic, but that in higher urban densities, it has the potential to become pneumonically transmitted human to human? Or is there another mechanism that is also preventing more pneumonic transmission of the plague?
So the more than 80% of the case recorded in Madagascar are of a bubonic form. So when people are treated on time, they may not develop the pneumonic form, meaning that early treatment with antibiotics can really stop the spread.
Yeah.
But because plague epidemics are most of the time happening in remote villages, it is difficult for people to get the help they need, or sometimes they choose not to go to the hospital.
Yes.
And what happened in 2017 was somebody traveling from place to place. So the person was already sick in the car, like a public transportation, developing the symptoms of pneumonic plague, and then spreading it to people inside the same car, and those people in turn spread the disease wherever they went.
And I guess this falls into sort of a third category of plague, a sort of septicemic variety of the plague, just through contact with bodily fluids or just with the contact with another person infected carrier of the plague.
Yeah, that's also another form of plague, but quite difficult to diagnose. So maybe that explains the really low rate of report of septicemic plague.
And how common is it to have silent carriers of the plague? Is Yersinia pestis virulent enough that once infected, even with single counts, that you will go on to develop symptoms of the plague unless treated, or do some develop natural immunity and show no symptoms, outward symptoms of the plague?
As far as I know, in humans, the human system is really vulnerable to the attack of Yersinia pestis. So, in my opinion, it's really unlikely for someone to get infected and remain infected but not developing symptoms.
Yeah.
But honestly, I don't know. I don't think I have enough knowledge of the human plague to answer that. But in wild animals, there are the natural carriers, and the cycle of transmission is often comporting a component of animals that are resistant to plague infection, but potentially when they meet other animals that are sensitive, they may infect those animals and provoke large zoonotic transmission.
And so, across a scope of Madagascar, do you find therefore differences in susceptibility, but also in morbidity of carriers? So, particularly in rats and mice across Madagascar that are more sensitive or suffer higher mortality when infected with the plague than other parts of Madagascar.
In humans?
In wild animals like rats.
So, in humans, it's hard to say. I think unless somebody is doing a study of the available database from spanning from, I don't know, 30 or 40 years and including different region of Madagascar. As far as I know, nobody ever did that kind of work. But in Madagascar, there are two reservoirs of the Yersinia pestis, the black rat, called also the Rattus rattus, and the sewer rat, Rattus norvegicus. And studies showed that Rattus norvegicus, the sewer rat, is more resistant to Yersinia pestis infection. When we talk about resistance here, is like the lethal dose, like the lethal number of bacteria that can kill an animal. And that's really important in terms of plague ecology, because the distribution of those two species of rats are different. The black rat, the susceptible one, is more from the countryside, from the villages. But the more a place is urbanized, the sewer rat is taking over. And that may explain why the current state of plague in Madagascar is that it's not really happening in those populated urban area, even if there are a lot of rats. So the rats are more resistant, the sewer rats, when compared to the black ones. But it doesn't mean that there is no risk, because rats are also moving with people and goods. So it may happen that a rat from the countryside may come to town, and things may happen also, like surveillance of rodents showed that in those urban areas, the rodents are still exposed to Yersinia pestis, meaning that they developed the immunity against Yersinia pestis.
And effectively become silent carriers of Yersinia pestis. Or partially silent, at least.
Yeah, well, we don't really know if at some point they are, if they seroconverted, it means that at some point they were exposed to the bacterium, but they didn't die.
Yeah.
But then after, does it mean that they are silent carriers?
True. I guess it can be quite hard to measure the true morbidity, or I guess the daily adjusted life years in rats.
Yeah, one thing that was always assumed to be like an indicator of plague is rat fall, meaning people reporting unusual death of rats. But that is not very common in Madagascar.
And of course, I guess contact with dead rats or with other dead mammals, including lemurs, is one of the major sources of transmission of plague. Is this also true in, presumably not as much so in Madagascar? Then I know in the States, contact with dead animal material is a major source of most sporadic cases of plague.
Did you mention lemurs
Yes.
earlier?
So I mentioned dead lemurs in contact.
So far, I don't think lemurs are involved
Okay.
in any like zoonotic disease transmission to human. But of course, contact with a dead rat is always risky, and people are not really encouraged to try to control rats using those snapping traps, because when the rat is dying, the fleas are spreading, and the fleas are going to find a host anyway.
Yeah.
So the person in the household using that kind of trap, the people are at risk.
So maybe moving on to fleas specifically, and some of your in your work, and I think you also recently awarded a centennial travel scholarship to explore vector transmission in plague, exploring fleas specifically, the Pulex genus more broadly. Could you, in terms of flea species in Madagascar, and why the flea is such a capable insect in terms of transmitting the plague, could you maybe describe that more?
So fleas are blood feeders, meaning that they are strict blood feeders. They don't rely on other type of diet to survive. Not like mosquitoes, that only the females rely on blood to produce the eggs. But male and female fleas, they feed on blood. And they also have like an interesting structure in the digestive tract that may favor the development of Yersinia pestis, forming a biofilm inside the gut. So the biofilm is important for bacteria to grow and to resist to the harsh condition inside a digestive tract. So I think the evolution of Yersinia, if I may say, took advantage of that structure in the flea gut to be able to be transmitted from a host to another. But the fleas are also suffering from the infection, if we can say suffering, because they die from the infection, which can totally block the digestive system. So many species of flea, when they are infected, their lifespan is shortened by the infection. But not all the fleas can develop that sort of blockage because the structure of the guts are different from species to another. And also the biology and the physiology of fleas can be different also between species. Some fleas are, let's say, frequent feeders. They ingest small blood, meal, and feed several times. Other can also ingest huge amount of blood and still feed a lot. And some other are just feeding once or twice a week and then be happy with that. So even these different habits, let's say, can influence the growth of Yersinia pestis in the gut and then make it less or more or less a good vector. For Pulex irritans, the species I am studying currently, very few things are known about that flea. But what I found quite interesting about Pulex in Madagascar particularly is that this is the flea in the household, meaning that if you go in the rural area of Madagascar and if you hang out inside people's home, you will probably get out with one or two fleas.
Yeah.
In many parts of Africa and even in the United States, flea infestation means the cat flea, Ctenocephalides felis. It may be associated with the presence of pets like cats and dogs, but in Madagascar, as far as my samplings are going, I confirm that Pulex irritans is infesting homes. But Pulex irritans is not taken seriously because, that high number with plague in the area, people may think that if Pulex irritans is a vector, it should have more cases than what we observe nowadays. But we cannot rule out the possibility with that huge number. Can you imagine a room, a bedroom, and we sample one night and we can catch around 500 fleas in there? And that's just like 8 hours or 6 hours of sampling. So can you imagine that kind of environment? And if somebody is infected in that room, what would be the risk for the entire village? So even if Pulex irritans is not a famous vector, I don't think we should just rule out the possibility of it playing a somewhat role in the transmission. And it has been found infected during epidemics too. So now I am studying the factors making a household prone to infestation, like why some households have 500 and why others have only 20 and others have none in the same village. So if we can solve that already, we can know what are the risk factors for those people, and maybe help also to inform some sort of control by the people themselves.
And do you imagine, and interested in particular mechanisms that promote, for example, Pulex irritans, and where so both their own reproduction, but also where they happen to migrate outside of reproduction, is in terms of the size of the village? Do you imagine that their location relative, for example, to large bodies of water, or availability, simply density, that different houses will have different densities of people within those houses? What factors do you think are relevant in actually influencing the density of Pulex at any one time?
So from my entomology training, so I am more, my reasoning is starting from the biology of the insect itself. So for me to understand what are the factors, I'm starting to look at what are needed, what conditions are needed for a flea to survive first and then to thrive. So flea, in their life cycles, they lay eggs, and those eggs are making larvae, and those larvae needs particular conditions, temperature and humidity, to survive. And what's most of the time is happening, people try to get rid of the adults because the adults are the one bothering them and biting them. But if nothing is done to prevent the eggs from hatching or to prevent the adults, the larvae to make adults, it's just quite a waste of time. So for me, I'm looking at the right condition for larvae to thrive in a household, and larvae are growing on the floor, on inside cracks and crevices.
Yeah.
They are really doing well if there are a lot of organic matter in their environment. So they feed on various debris. They need humidity and heat. So all that condition can be found in certain homes. Something that was striking from my data analysis so far is the nature of the flooring in those households. So fleas are thriving within households with dirt floor covered with mat. So these are traditional mat made of plant fiber. They are quite
Like
beautiful.
palm fiber or?
Yes.
Yeah.
But even if it is very clean on top of it, people cannot really clean underneath it. And I think flea immature stages are living underneath it. So we didn't find any flea infestation, like high rate of infestation in households with concrete and board floor when compared to the households with those mat floor. So that's one factor.
Even given enough circulation with the outside, still ultimately it was very much centered on the difference in material of the flooring.
Yeah, so that's one thing, but there is also like the movement of the adults.
Yeah.
And we don't really know about that. It's really difficult to track like where the fleas are going. But I think there is always a possibility of reinfestation or exchange between neighboring homes or between people, meaning that a household located next to an infested one, it will be always exposed to potential of reinfestation if people go to each other's home. Something very frequent in Madagascar. People like to hang out in each other's home. There is also big gatherings. So we don't have any evidence from data about this, but what I'm going to say here is just from conversation with people in the village. So they noticed that after big gatherings, such as like parties, or wake when someone is dying, the annoyance from flea is really high.
Yeah.
So
truly irritants, yes.
yeah, probably meaning that everyone coming to those gatherings are somehow carrying their fleas and put it together in the same place. It happened to us also sampling in a house where the family gathering happened before. So we got like an incredible number, and after that we asked them like what happened to this house, like is there something special? And they mentioned, yes, we had like a family gathering 2 days ago, and you guys just arrived at the end of a family gathering. So yeah, but it's very difficult to, capture all of those moments. You never know when people will have their party or family gathering.
I guess it could even occur with smaller gatherings where you meet only a few individuals, and that's an opportunity for potential spread of shed fleas as well.
And we learn from ourselves. I mean, my team and I, we can't help but we also have fleas on us. So once we start going into people's home, talking to them or sampling, we also have some on us, even if we were freed of flea at the start of the field work. So it means that those flea can like move around with people.
And so I guess this is intriguing because I know that Yersinia pestis, of course, can also be found in soil. Is that do fleas ever acquire Yersinia pestis freshly from soil, particularly in housing that perhaps has exposed soil, or do you think that most cases of newly infected fleas arises just through transmission from another host?
So the only known transmission, like the way of infection of a flea, is through blood feeding, meaning that a flea needs to feed on a host, and that host needs to be at a certain septicemic level to be able to infect a flea.
Yeah.
So I think exposure to infected soil is less likely to infect a flea. But there is discussion around the potential of infected soil to infect rodents. So that's quite interesting, because of the big question for all plague study is like, where does the bacterium hiding during those quiet times where there is no episodic, no human cases, but it's still there, but we don't know where it is.
Yeah.
So there are a lot of, ideas within the group of scientists working on plague. There is the hypothesis of infection from soil, as you mentioned. So the Yersinia might be engulfed within like a microorganism, and those microorganisms thriving in damp soils, and later rodents might come into contact. I know it's kind of far-fetched, but we cannot...
Like a biomagnification sort of effect, yeah.
There is also the hypothesis of flea as reservoir, because of the lifespan, because also adult flea that are infected are let's say expelling feces containing a high amount of bacteria, so their feces are infected, and the larvae are feeding on adult feces too. So there are a lot of things to be explored in there, and the reservoir, like mammal reservoir too. I don't know if it's possible for a mammal to remain infected during a long time and potentially transmitting to another mammal that is also resistant, and so on and so on. Like that mammal might heal over time from the infection, but already passed it to another one, and so on. So, and you earlier mentioned also like human reservoir. We don't really know about those, but there are people seroconverted also, but never showed any symptoms of bubonic plague.
Yeah, which is quite, I think, quite a scary prospect as well. We know this, of course, for typhoid fever as well. There are some who never show symptoms of typhoid, but are still able to pass it on to others in that carrier state. I was wondering whether you could describe it in more depth, how the sampling methods you use, and some of the molecular techniques you use to actually identify, firstly identify maybe strains of Yersinia pestis, and other ways you catalogue both in maybe infectivity, but also just catalogue different species of pulex.
So So far, I'm not really working on like molecular methods to identify pulex. I'm more like of an old school, like using microscope
Oh yeah.
to identify the species. But I know that there is another like sister species that might be confusing. But so far in Madagascar, we only have Pulex irritans, but here in the United States, there is also Pulex similans. For Yersinia pestis detection, we use polymerase chain reaction, in fact, to detect gene from the bacterium in fleas, in mammal samples, and also that is also used as a diagnostic tool for human in Madagascar.
Yes. And are there, I guess it links to the previous question, so of Yersinia pestis, do you find any variety in different strains of Yersinia pestis that's either that carry different morbidities or that are just otherwise found in different villages across Madagascar?
Yeah, I think there are scientists working on that, like the variability of Yersinia pestis strains, and it's really interesting to see how those different strains are spreading all over Madagascar. And while some strains are mostly found in those like silent focus, but it does not imply that it is less virulent than the other strains.
I guess it's just a matter of differently evolving.
Yeah, like
strains, yes.
evolution.
Yeah.
But it's quite interesting also to understand like where is, for example, the source of the infection and where it spread, like spatially in the space in Madagascar. For example, it all started in these small villages, and then this strain moved to like 500 kilometers away, and then stay there for like 10 years, and then move to another place. It's always interesting to me, even if I'm not personally working on like the evolution or the genetic of Yersinia.
Yeah, I think it's quite interesting just to track out almost the chronology of how a particular strain has moved around as it's been carried and transmitted. Maybe I would be interested also just in exploring, particularly because you mentioned there's sometimes resistance in accepting treatment for some common plague treatments, so particularly taking antibiotics to prevent the more severe pneumonic form of plague. How have you found maybe some of the existing Malagasy cultural values and your work as a scientist, as you come to it with almost two frames of mind, perhaps? How do you find the two intersect?
Yeah, that's quite a delicate question, I think, but it's still interesting. I know that it's very common in Madagascar to wait until last minute to go to find a doctor, because there are, I think, many reasons to that. First of all is the financial reason, like people are expecting expenses, so if it can disappear somehow, well, you don't have to spend that money on medication.
Yeah.
And there is also the fear of just going to a medical facility, because a lot of people in Madagascar did not receive like higher education. People sometimes feel like they are vulnerable in front of like healthcare personnel. They don't know what they are talking about, and it's just a bit might be annoying for them to interact with people they don't really know. Or there are people totally in denial. Like they don't believe that plague does exist, and they cannot believe that it may happen to them or their family member, so they just resist and like, no, this is not plague.
Yeah.
Yeah, and I think part of it was also some measures taken during colonial era about bubonic plague, because let's not forget that plague was a disease from outside of Madagascar, introduced a little time, like late 1990s. So plague came into Madagascar with a steamboat during the third pandemic. So that's at the same time started a few decades earlier, the colons started to arrive. So I think in the collective mind, plague is also a disease carried with the colons, even if we know that nobody might be wished to bring plague with them.
Yeah.
But it happened, and so the colons, to prevent the spread of the disease, took really harsh measures. For example, the isolation of people, the destruction of houses. So for Malagasy people, it's also very important to follow all the rights around death and afterlife. So some of the measures that needed to be taken in order to prevent the spread of disease are directly against the wake, for example, which is a very important ritual in Madagascar, even if it may put a lot of people in danger, particularly if it's a pneumonic plague.
Yeah.
So people's gathering, like touching also dead body, and their relatives. So sometimes also another measure taken is to bury the deceased outside of the family tomb. So that's a very painful kind of loss. So there is the loss of a beloved person, and the beloved person cannot be with the entire family also within the family tomb. So that's another tragedy. So that may also explain why people are resisting and like recognizing that this is plague, because they just want to avoid like all of those measures. But those measures are necessary to try to stop the spread of a disease. So it's always like a hard decision to tell people like what to do for public health sake.
Yes. And I can imagine, of course, it may even perhaps cause resentment, and there aren't that many generations between the first event, as you described, and now, and maybe that resentment, colonial resentment, is still present, even if much of the plague now is now it's sort of historically rooted now in Madagascar.
I think there is also like a lack of trust between the population and the healthcare. It's sometimes, where do you find the logic when somebody's sick, they are waiting last minute to go to the hospital, and then they die when they arrive at the healthcare facility, even if the doctors are doing their best to take care of them.
Yeah.
And then the family is assuming that those people are, killed at the hospital because the doctors don't want the disease to spread. So everyone diagnosed with plague, they would just like killed at the hospital.
Yeah.
That's a difficult situation. I'm sure that the doctors are also going through really rough time, like dealing with somebody, like a village with plague, and the insecurity. So a big kudos for the doctors and nurses working in those villages. I know it's not easy.
Because it's particularly challenging because once they've reached that stage to the very last point at which medical attention could be given, they are potentially also the most infectious and contagious stage of the disease, and so they are placing themselves on the front line. So it does quite a difficult environment, both for the family, but for medical staff, and obviously for the patients themselves.
And that's why I think even if plague is just now like touching 400 or 200 people within a land populated by 23 million people, I think it's always important toward working on like helping to lower even the number. Like nobody should die from plague anymore in Madagascar or anywhere else in the world. But I think it's not only a matter of scientific research. There are also a lot of like societal factors that are favoring the persistence of the disease. But the more we know about the mechanisms of the disease, I believe that the more we are able to reduce the number, even if we cannot really change the reality of those people.
Yeah, it would be tempting to discuss also more in closer depth exactly how much traction you think directly treating the symptoms, and obviously the risk of antibiotic resistance is also present. But I was maybe interested in also moving on to some of your work, particularly supporting the ICALA STEM innovation and women in STEM, and maybe wanted to first learn more about the foundation and also your involvement with them.
Oh, so ICALA STEM, it's an association of obviously women in STEM in Madagascar. So ICALA is a Malagasy word to describe like a girl. The association is promoting the profile of Malagasy women working in the STEM domain. So I started to be part of this association back in 2017 when the association just started, was very young, like 1 year old. Yeah, and I remember we were like 30 or 40 people at that time, but now we are like more than 300 and scattered all around the world. So we have different chapters like in Asia, in Europe, here in Americas, in Africa, and in Madagascar, of course. And we are all working together, and our effort is particularly to promote science in Madagascar and education.
And do you feel that this is a part of the goal is to allow to promote individuals women in science in Madagascar, or to promote women in science, whether that's in Madagascar or anywhere else in the world, to providing grants to travel abroad to engage in science research abroad?
I think first thing is to provide like a role model, a reachable role model, someone that every children or a future scientist in Madagascar can identify with, meaning that they would look upon their role model and say like, hey, I can do this too. I'm a girl from a village. She was a girl from the village too, and she did this and that. And we will probably go through the same challenge and hardship, but hey, we can do this. So that's, for me, is a very important message vehiculated by the members of Icalastem. So we are all there. Some of us are really eminent scientists in their field. So we have astronomers, we have physicists, we have physicians,
Entomologists.
we have entomologists too. We have conservationists. So I think that's a very strong message to the women and future scientists from Madagascar. And it's also a platform for us already within the association where we can talk to each other, exchange ideas, and even, like the friendship within the association is just amazing. Like, for example, I am here in the United States and I can talk to another Malagasy woman scientist working in Japan.
Yeah.
Like, outside of the association, what are the odds for me to talk to that person? So I think the existence of that kind of friendship is just amazing. And also our work trying also to boost, let's say, the career of students in Madagascar. The goal is not always to give grants and make them come here. But if they can just achieve things in Madagascar, that would be great too. Even finding money to print manuscript and books, sometimes it's really challenging in Madagascar. And also having access to resources such as internet, even electricity is challenging in certain places. So there are the challenges, but there are also brilliant people in there, and there are also the ones willing to help. So all of this is making Icalastem, I think, a great platform to help the next generation and also the current generation not to give up.
Yeah.